Another to another edition of the CanNRT Research Forum series. Today's forum is titled Good Trials Only, Integrating Evidence to Improve Outcomes for Children and Families. My name is Eliana and I'm the Training Program Manager at the Canadian Neurodevelopmental Research Training Platform, or what we like to call CanNRT. So CanNRT is a collaborative training environment that supports research excellence and sustainable and equitable pathways for early career researchers in neurodevelopmental conditions across Canada through multidisciplinary cross-sector learning, mentorship, and connectivity. And one way that we achieve this is by bringing the CanNRT community together in such forums, which showcase the diverse work of our members and promote collaboration and knowledge exchange on topics related to neurodevelopment and associated conditions. I'd like to begin by acknowledging that CanNRT headquarters are located on the traditional territory of the Haudenosaunee and Anishinaabe nations. This land has long been a place of meeting and exchange between Indigenous nations, including the St. Lawrence Iroquoians, the Huron-Wendat, and others. I would now like to introduce today's moderator, Ms. Kelly D'Souza, a master's student in educational psychology at McGill University, specializing in inclusive research and inclusive practice and research, who will guide us through the forum and introduce our distinguished speaker. So Kelly, please take it away.
Thank you, Eliana. Good afternoon, everyone. It is my pleasure to introduce Dr. Lauren Kelly, Associate Professor in the Department of Pharmacology and Therapeutics at the University of Manitoba. Dr. Kelly is a certified clinical research professional and serves as the Clinical Trials Director at the Children's Hospital Research Institute of Manitoba. She completed a PhD in pharmacology and toxicology at the University of Western Ontario, followed by 2 postdoctoral fellowships at Mount Sinai and the Hospital for Sick Children. Dr. Kelly is the Principal Investigator of the Clinical Pharmacology Lab, where her research focuses on evaluating drug safety and effectiveness in children, as well as during pregnancy and lactation. She also serves as a scientific director for the Canadian Childhood Cannabinoid Clinical Trials Program, where she leads several clinical trials examining the use of cannabinoids in pediatric populations. Finally, and perhaps most relevant in today's session, Dr. Kelly is the Director of Impact, a national training platform designed to increase capacity for maternal and pediatric clinical trials. So please join me in welcoming Dr. Lauren Kelly.
Thank you, Kelly, and thank you, Eliana, for the invite. Kelly, if you can just confirm that everything is good and you can see my slides.
Yes, I can see it.
Excellent. So thank you for the introduction. Today's talk is really going to be an introductory talk for those of you who may be new to the clinical trials world. And we're going to talk today about good trials and good trials only. So before we begin, I just wanted to acknowledge that where I am currently located, I have the privilege of living here currently in Winnipeg and raising my family on the beautiful Treaty 1 territory, which are the homelands of the Anishinaabe, the Cree, the Dakota, the Dene, and other Indigenous peoples, and the national homeland of the Red River Métis. A little bit about you for those of you who haven't met me before. So I'm originally from Ontario and I've been here in Winnipeg since 2017. I'm now an associate professor in pharmacology and therapeutics. I have 2 tiny humans and 1 furry one. And as Kelly mentioned, I'm the principal investigator on 3 clinical trials. So I run both an active research program. I've also experienced clinical trials as a participant and spent several years in a clinical trial. And I have the privilege and honor of directing one of our national training initiatives to help support the next generation in learning about the design, conduct, and operations of clinical trials. And I'll talk a little bit more about impact today. As I mentioned, this is really an introductory talk. I am happy to meet with individuals at a later point, either one-on-one or in a group environment, to discuss sort of more specific methods questions. But our learning objectives today are really to understand and define key terminology related to clinical trials and oversight in Canada. To understand what the purpose of clinical trials is and what makes a good clinical trial in terms of its design, and to help identify some opportunities and resources to learn more about clinical trials in case this is a methodology that you'd like to use throughout your research career. So getting started on the first objective. So clinical trials are a type of clinical research method. So Some of you may have worked in clinical research before, part in a cross-sectional study or a prospective study, retrospective study, case control study. These are other different kinds of study designs that either look at a present moment in time, follow people forward in time, look backwards after an event has happened to try and identify what might have caused that event. Clinical trials are always going to be prospective. So they involve designing a study, consenting folks, and always following them forward. So clinical trials are prospective studies that assign an intervention to human participants under study conditions. What this means is that clinical trial will determine what someone is taking and how much they're taking it, how often they're taking it, and what kind of data they're collecting. And they must follow a pre-planned study protocol to systematically measure and analyze an outcome in order to answer that question about whether something works and is safe. There are many definitions of a clinical trial. So Health Canada has a very specific, narrow definition that is sort of specific to drug trials. But I like the first definition on the screen here, which is what is included in the Tri-Council policy statement, which governs all of our Tri-Council funded research here in Canada. And this really aligns with the World Health Organization definition as well. And this states that any research study that is prospectively assigning human participants or a group of human participants to one or more health-related intervention to evaluate the effects on health outcomes. So trials can look very different, and this depends on what we know about the intervention, where the trial is being conducted, what's known about the condition to date. Trials can be led by investigators. So what I do is investigator-initiated trials, meaning that myself and my institution, in this case, the University of Manitoba, are the sponsor for the clinical trial. Or clinical trials can be sponsored by industry or companies that are investing in developing a product. Clinical trials happen in single sites at one place or can happen in multiple centers, multiple countries. There's no limit on how big a clinical trial can be. There's also kind of no limit on how small they can be. So they could be a single arm. Maybe it's just looking at if something is feasible or if something is safe. Or they could have many arms if there's lots of different things that they want to look at. They can be looking to confirm if something is acceptable and safe. They may have individuals serving as their own controls. It may have a different control group. And all of this to say that a clinical trial looks different depending on the question that it's trying to answer. And so because this is an introductory talk, I just wanted to start with a couple of key terms before we jump too far into the session today. So the intervention. An intervention is essentially whatever is being studied in the clinical trial. So this could be a new educational curriculum on how to teach grade 3's math. This could be a new drug that's coming to market. It could be a new AI-assisted technology to help grade seizure severity. Whatever that is being studied is your intervention. Your control or comparator is what are you comparing that intervention to? Typically, this could look like either a placebo, it could be to no treatment, it could be to whatever standard of care, or what the participant would receive if they weren't in the clinical trial. Outcome, the word outcome means what are we measuring to assess the change related to the intervention? So how are we actually going to know if this intervention was different than the control? What are we going to measure to say, yes, this was better, or no, this was worse? And that also includes how we're going to measure it. So what's the tool we're going to use to measure that outcome? And what's the time point at which we're going to be looking at whether or not a change happened? So it's really important to define this ahead of time in clinical trials as what change do we expect to see and what is going to make this intervention, what change do we need to have in this trial so that folks believe this intervention is something they should adopt. So the goal of a clinical trial, remember, is not just to generate data in a publication, but to actually inform how intervention may be usable in the real world. And so we need to think about, okay, well, based on what we know in this condition and what's currently available for this for whatever it is, whatever problem we're trying to solve, how much change do we need to see? And it might be a lot. It might just be a little though. And so it's really important to think about that before we start a clinical trial. And the word systematic, when we think about clinical trial, means that it implies an organized multi-step approach. Clinical trials are not something that just happen by random chance. The trial design is not random. So when we hear the word randomization, it's not that, there's any random component to how trials are designed. It is a very systematic approach that is taken, and that's what we're going to talk a bit more about today. The term randomization. So randomization means the process of how we allocate participants either to an intervention arm or to a control arm. There's lots of different kind of concepts around randomization. So I just wanted to define a few key terms in case you're reading a publication and you're thinking, what does it mean if it's block randomized or if that randomization is stratified? So randomization, a block just means the number of participants you're going to randomize before they're equally to both groups. So what I mean by that is, you might be A, B, B, A, B. Maybe that's 4 participants, maybe it's 6 participants, maybe it's 10 participants, so that you know that they're equally allocated per group. So typically in many trials that have block randomization, they would use a random block size. So alternating how many people, they make sure that those groups are balanced. And stratified randomization means that there's something that we know is going to affect the outcome, meaning that could be age, it could be sex, it could be a specific disease severity. And we want to make sure that the people with that factor, so let's say disease severity, we want to make sure people who are really sick and people who are less sick are equal in the treatment group, in the intervention group, and the control. So you can do that by what's called stratified randomization, meaning that that would be a factor you would balance between the groups when you're assigning them to either get the intervention or control. Randomization can also be done at the level of an individual. So this is probably what's most common and what people most think about when they think about clinical trials. It can also happen at the level of a group. So for example, cluster randomization is when a group is actually randomized either to the intervention or control as opposed to an individual participant. And this is really helpful when we think about things like public health interventions, where whatever we're doing is going to be done at a population level. So if we think about anti-smoking ads on the radio in your, the car radio, well, you can't deliver that to one person. You can't control who listens to the radio, but you could regionally, radio stations don't reach everywhere, so you could allocate groups to receive an intervention or not. And the third way you can randomize is also in which the order in which treatments are received. So for example, if you had an N of 1 clinical trial, they may be their own control. So they may receive 1, 2, 3 different active arms and a control arm. And you can randomize the order or the sequence in which they receive those different interventions. So lots of different ways to randomize when it comes to clinical trials, but the design itself is never random and always informed by a multi-step process. Another key term I just wanted to talk about before we move on is this concept of blinding. We're not going to talk too much more about it in this talk today. But blinding means that the participant or the party, because it could be the investigator, it could be your data analyst, it could be anyone who's assessing an outcome, is unaware if the person in front of them was assigned the intervention or the control arm. And this is done because we want to prevent people's expectations from influencing their outcome. So oftentimes folks can maybe perceive that they're feeling a little bit better if they know that they were getting the drug, and we would like to obviously try and avoid having that bias. So that's why blinding is done. So clinical trials happen at lots of different, one place or many places. They can be sponsored by industry or by individuals. They all must be overseen by an ethics review board in Canada. They must be conducted by experienced and multidisciplinary teams. They must all disclose funding sources and any conflicts of interest. They must provide participants with the opportunity to provide informed consent. And what informed consent means that those folks who are in the clinical trial have not only enough information about the study, so what's being asked of them, how many times do they have to come to clinic, what forms do they need to complete, how many blood draws do they need to do, but also what any risks are and any protections that are in place for their safety and also for the data integrity and their privacy. Providing informed consent means they know all of these things and they've had an opportunity to ask questions before they say, yes, I'd like to be involved in this study. Clinical trials have to have data integrity plans, so they must all be registered, meaning that before they start, they say in a public database, I am going to do XYZ in this study. And the reason we do this is so that when folks report the trial at the end of the day, they have to report XYZ that they said they were doing in the study. So ideally, studies are going to report what they said they were doing and not just, the finding that may be most significant or most headline grabbing, if you will. And we want trials to all have timely reporting, meaning whether the trial succeeded, whether it never got off the ground, we need trialists to all be accountable for what happens in the trial. And they should all have a plan in place for safety monitoring. And we'll talk a bit about when Health Canada is involved in those decisions and what looks different if you're doing what we call a regulated trial. But when we think about, people ask me, well, how do trials happen then? If trials aren't random, how do they get started? And the reality is that there has to be some kind of problem, either an opportunity, there needs to be an idea that many people are behind. So that step two of forming a multidisciplinary team is a crucial part of vetting if that idea is suited to a clinical trial. So is a clinical trial the right tool to answer this problem? And that is something that is best answered with a group of folks who have different perspectives. And the next thing is to understand, what do we already know about this problem? So what do we know from the literature? What's already been studied? What do we know from patients and families? What are their priorities in this problem? And what do clinical colleagues and research teams need to change in order to be able to improve things? And so those three pieces are critical before you can really convince someone that your question is important and your team is qualified and that you are the right group to actually answer this question with a clinical trial. And I'll talk about impact a little bit more at the end of this session, but there are many things that you have to know before you can convince me as a peer reviewer that you are ready to do a clinical trial. And so these are kinds of, we have lots of tools now that we've created to help our fellows walk through, what do I actually need to think about before I am ready to propose a clinical trial? And so I'm happy to talk more about this a little bit later. But the most important thing to start with is clearly defining what is it that the problem, what is the problem? So who does it affecting and what is the population that you're going to include in your trial and not include? So are there folks where this may not be safe to study? Are there folks where we maybe already have an answer for a subgroup in a larger context? What is it that you're testing in the trial? So if it's a drug, well, how much of it are you going to give? How often? Where are you going to source it from? What are you going to compare it to? And when you select a control arm, you're ideally going to be comparing to what is happening outside of a trial. So if this patient was to walk into the clinic, what would they get for their disease? Or what happens at a school that isn't running this new math teaching curriculum? That should typically be what you think about for your control group. The outcome, again, like we talked about, is what you're going to measure to demonstrate that there is change between that intervention and that outcome. And the timing, or when are you collecting the data, and the primary analysis time point. And so we want to make sure that questions are meaningful, but we also want to make sure that they're informed by multiple perspectives. And so when it comes to thinking about someone in a trial, so a participant in a clinical trial, what does it look like for them to get involved? How do they get involved in clinical trials? And the reality is it's a very diverse and complicated and sometimes lengthy process. So they first have to have a diagnosis. So as we know, especially in the case of NDDs, that diagnostic journey can take many, many years. And they may actually need a formal diagnosis to be eligible for the clinical trial. Especially if it's a clinical trial that's being conducted by industry for the purpose of drug marketing, they will likely have very strict inclusion criteria around having a formal diagnosis. Then they have to be referred to a trial team. So maybe the physician that cares for them knows about the clinical trial, is involved in a clinical trial. Amazing. But that is many times not the case. So they would then need to have identify if there is a process for them to be referred into the clinical trial. And again, this does not exist in most trials. They also then have to be eligible. So eligibility is different for every trial, and it may include things like being able to speak a specific language, being able to attend clinic on specific dates, a specific number of times. Lots of those eligibility criteria screen out potentially eligible participants, not necessarily for a safety or efficacy standpoint. They need to provide informed consent. So again, this is where they've already, kind of met the criteria. They said they're interested in research. They look like they're going to be a good fit for the study. Now we can explain the study to them, answer questions, go over any risks or benefits, any compensation, privacy, all of those things before they would be enrolled and randomized. And once they are allocated, they either would get, an intervention or a control. Let's say in this example, they would go through a variety of study-related activities. These range from trial to trial. They could all take place at home. They could all take place in clinic. They could include blood draws, no blood draws. There is no set number of activities that are required in a trial. But at some time point, we're going to measure an outcome and that's going to tell us the answer to our question of, is there a difference between these groups? And then we're going to end the study and we're going to share our findings. And from an investigator's perspective, so if we kind of swap sides and think about from those of us who plan trials and those of us who lead trials, what does this look like? The planning and design phase should probably just be a orange bar that covers the whole page. This can take many, many years. This is the phase where you are generating evidence and consensus and a network that is going to convince folks to give you money to do your trial. So you have to get funded, you have to get all your approvals in place, and then you can start consenting and recruiting and following up with your trial. And at the end, of course, doing your analysis and your reporting and your knowledge mobilization. The trial process takes many years. And if we look at a real example, so this is my first clinical trial as an investigator. This trial was funded by the Sick Kids Foundation, part of an early career award, in 2019. And we just had our last data safety monitoring board meeting to close out our trial in March of 2026. This is a pilot trial that ended up recruiting 3 participants and took this many years. So all of this to say, it is a very lengthy process, and there are lots of rules. Who makes these rules? So it depends on what kind of clinical trial you are doing. So that checklist I went over, that's applicable regardless of if you're studying a drug or a new behavioral approach or an educational pathway. If you are studying what is called a regulated trial, you must follow good clinical practice. And this is guidelines that are set by what's called the International Council for Harmonization of Technical Requirements for Pharmaceuticals for Human Use. A very long name. Everyone calls them ICH for this reason. And ICH makes the rules internationally. These rules are then adopted by the FDA, the European Medicines Agency, Health Canada, the PMDA in Japan, the MRN in Australia, etc. There are many, 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 many guidelines that I will not be talking about today. I do, however, want to talk about the good clinical practice guidelines. So there are 13 principles of good clinical practice. I am not going to read through them all on the slides, but what I want everyone to understand is that these are international codes of conduct. These apply to all trials that are regulated within this space, regardless of what country you are conducting them in. And the purpose of good clinical practice is to ensure that the data that is generated is good quality and is auditable, meaning if a pharmaceutical company is conducting a study, the government needs to be sure that if they want to, they can come and look at the data and understand where that data was sourced from. So how did they decide the liver enzymes weren't high for these participants? Where were they getting the clinical information? So the purpose of these guidelines is to make sure that data is not only collected in a good way, but also can be verified for the purposes of a marketing authorization. And all of this to say that they do not apply to all study types. So the term regulated clinical trial is not necessarily a formalized Webster definition, but more of a loose term for what we talk about, at least here, as a trial that would need to be submitted to Health Canada. So when I say, all trials must have ethics, all trials must be registered, that is true for every kind of intervention. Regulated trials are those that require you to submit a clinical trial application. Those are the ones where you must follow good clinical practice. And these are the trials that can be assessed through Health Canada's Good Clinical Practice Inspectorate program. Part of what Health Canada does is approve clinical trials, so approve the product for sale under the controlled environment. They also have the authority to inspect. And what that means is for regulated trials, they have an inspector program. They will come to your site. They will look through all your clinical records. They will verify that what went into your electronic system matches the paper system and all your signatures are in place. And that inspectorate function is only applicable, again, to trials that are regulated. So what is regulated? So anything is regulated if it is not yet available for sale or is being used outside of its approved indication. And this is the big challenge for those of us who work with kids. Most of the drugs that these children are receiving, and especially when we think about NDDs and, for example, kids with autism, there is no approved therapies in Canada for children with autism to help support core symptoms. So those are all medicines that are used off-label, meaning even if it is a standard practice, standard of care drug that, 80% of developmental pediatricians are using for that indication, if it is not on the DIN, it is not considered, meaning it falls in the regulated bucket when it really shouldn't, because it really is a standard of care use. It's a bit of a catch-22 here because it means then to answer questions about routinely used medicines that are off-label, you must follow the regulated pathway. And a regulated pathway means much more costs in terms of how long you have to store your documents. You have to have a monitoring plan. You have to have a lot more processes in place. So it does cost us a lot more to run a regulated trial. There is ways to know. So there is a drug product database and a medical device database called the MDAL here in Canada. So you can see on there what is approved, an approved use for that product in Canada. So again, that last point there, even if a drug is standard of care, if there's no labeling data available to Health Canada, it would be considered regulated. There's also not just one set of rules. So drugs, for example, fall under, so the Food and Drug Act, Division 5, or the Food and Drug Regulations, the FDR. Division 5 regulates drugs used outside of their label. There's also medical device regulations, biologic regulations, cell therapy regulations, specific things for cannabis products, and natural health product regulations. All of these are available on Health Canada's website. All of these are applicable if any of the interventions, so even if only one arm in your trial includes one of these, it will apply. So it's really important that as you're entering into a clinical trial, you are working with folks who understand the regulations and what regulations will apply in your setting. And there's some other terminology related to sort of regulated trials that I just wanted to sort of touch on really briefly around the role of a sponsor. So the sponsor or the principal investigator, these are the people who are overall responsible for the trial. They're responsible for the design of the trial, to get the trial funded, to report to the people who fund it, to do the regulatory submissions. So all our reporting to Health Canada, to the ethics boards, our safety reporting and oversight across all of the sites. This is sort of the boss, if you will, of the trial. And so if this person is a PhD like myself, we would be called a scientific officer. If they held an MD, they would be called a medical officer. And they're the person who is accountable for the trial integrity. A qualified investigator is sometimes a term called a site lead. We sometimes call them site leads. Each of the places where your trial is recruiting participants will have one qualified investigator. And this is in the regulation is they're sort of the site boss. So they're the main reports too for everyone who is being recruited at that site. So they make sure that the site is adherent to the protocol, that GCP is happening on site, that participants are safe. They must in Canada be a licensed physician or dentist. They are accountable again to the principal investigator. And then everyone else kind of falls into the sub-investigator category. So those are folks who support the QI. They may also be physicians who are recruiting for a trial. They may be pharmacists who are helping prepare intervention arms. Lots of different tasks can be delegated from qualified investigators. But it follows this very different structural hierarchy when you're working in a regulated environment in terms of the reports to and what needs to be reported in a specific time. So all of that to say is if you are working in the space of a regulated trial, there are different considerations and reporting structures that need to be embedded. So why do we need clinical trials and what makes a good one? So clinical trials play a critical component in our health system. So they help tell us what is safe and what works. They help look at new ways of administering or optimizing an intervention. So if we have a therapy that's given 7 times or 4 times a day and we can give it once a day, or something that's given by injection that we can give by mouth. They generate data for regulatory approvals. So again, all of that, getting a drug on market and getting that DIN is only possible with clinical trial data. So they do play a really important role in reimbursement assessments and determining out-of-pocket costs for patients and what they're able to access in terms of therapy. It's important because they help us understand who is benefiting and who may be more likely to experience harms and ways that we can prevent harms. And they also help us inform health outcomes, how we can optimize health outcomes in a controlled environment. And so when we look at clinical trials in terms of evidence-informed decision making, they fall in this category of we start at the bottom and look at case reports and expert opinion and move through our retrospective and observational designs. But really, when we think about evidence synthesis and what informs clinical practice guidelines, most of that is generated from clinical trials. And there's different types of clinical trials. So when we think about starting from the beginning, if we're answering a question, our first question is kind of, well, is it safe? Is it feasible? And that's what we would call a pilot study. And then we move to, does it work? Is this something that is effective? Is this something that is better than, and then we move to, okay, well, if it can work, is it better than what's already happening? And then we ask, does this work in the real world? And so when we think of like gold standard randomized clinical trials, that's really that phase 3 study of, is what I'm studying better than what's happening now? What's currently available for this specific population and this specific question? And the term comparative effectiveness, this is really that real world evidence. So in the phase 1, 2, and 3 trials, there's often very narrow parameters. Who can be in these studies? Who is, how many tests they need to do. And so phase 4 is almost more of what we think of what's happening in the real world. So in a larger or more broad population, do these effects actually hold and who is most likely to benefit from them? And we need more trials, and especially more trials that both include folks with NDDs, but also are focused on helping folks with NDDs manage symptoms, is because they ultimately are going to be what informs evidence-based care. And we know that families of children and adults with NDDs are accessing supportive treatments that are off-label. So, in the US, there are 2 medications approved for core symptoms in autism, neither of which is licensed in Canada. And off-label, again, means that we have to be studying them in the context of almost like a first-in-human infrastructure, which is really expensive and really onerous, and not most institutions don't have infrastructure for those kinds of trials. We need more trials because they affect reimbursement, and insurance coverage is often tied to what is on the label. So if we aren't updating our labels, then we aren't updating who can access a reimbursement for some of these therapies. And this just promotes variability in care, which is suboptimal. We need to understand who's most likely to benefit so that we can implement practice guidelines that include these recommendations. And this also makes trials more difficult because when there is no agreed upon sort of process or tiered approach to care, it also creates a lot of variability for a control group. Because if we say our control group is standard of care, is standard of care the same at all of the sites in our center is a different question, and one we need to think about during the design phase. And we need not only more trials for neurodevelopmental conditions, but also more neurodiverse people in trials in general. And so folks with neurodiversity and neural complexity also have other diseases. So, things like headache and pain and cancer. I mean, this all affects folks with neurodiversity. And so they're then being involved in trials helps ensure that the data generated is actually relevant to them and promotes evidence-informed care. And so we know kids and adults with neurodevelopmental disorders face different kinds of social and environmental factors in their life. And this can affect how they respond to an intervention. It can affect their ability to participate in research or access care. And so it's really important that their priorities are considered and that they're involved in informing how trials are done. And ultimately, clinical trials identify what treatments work best for which populations. And children and adults with NDDs should not be excluded from the societal benefit unless there truly is a reason why it's unsafe or irrelevant for them to participate. So what makes a good trial according to me? There's four things that I wanted to touch on in terms of what makes a good trial. So I've told you, why do we need them, and a little bit about how they happen, but what makes a good one? And so I like to think about these four concepts. So being well informed, meaning that it answers an important question that's well defined. It has to be trustworthy, meaning that I know when I'm reading it that they've attempted to minimize risks to participant and maximize the benefits. That it's generalizable so that its equitable access has been considered prospectively and that I understand who was in the study and if that reflects, the population that I'm trying to transfer the evidence from. And that it's reproducible so that it's transparently reported and statistically sound. And so just walking through each of these, and yes, that is angry Lauren trying to do paperwork, because I feel like that's often my job as a trialist is to do a lot of forms. But those forms are informed by multiple perspectives. And so when I say well informed, I mean considers the population perspective. So what do folks who are affected by this condition care about in a trial? What do they want to prioritize in terms of outcomes? What's important to them in terms of information that they receive back about their results. That includes scientific evidence. So, what do we already know from expert opinion, from retrospective studies, from observational studies, from other trials and similar conditions? And what do we know from clinical experience? So, what are people who are caring for this population experiencing in terms of risks and benefits, and how can we incorporate our trial into what's already happening in clinical care? And all of these perspectives come together with a trial methodologist, right? Someone who can take these perspectives and look at what are the regulations that we need to ensure this trial is informed by, and what are the methodology best practices. So how can we make sure that the outcomes we're selecting actually match what it is that we're trying to achieve? And in terms of trustworthy, I think this is one that often gets underestimated. I think, people, trialists have a bad reputation. There have been some folks who have done some very unethical trial practices. And I think, in order for folks to want to be in trials, they need to be confident that their safety is protected, that their benefits are being maximized, and their privacy is not going to be, data dumping all of their scores out into the internet or chat GPT. And that community, and a way to do that is by involving patient and community partners and having them ensure that your team is planning something that is minimally invasive and an inclusive in the design phase. And so I think it's really important that as we go into clinical trials, we think about how do we communicate all of the work that goes into ensuring trust in our trials. So how can we make sure people are confident that our study is aligning with ethical frameworks, with community frameworks? If we are working in community, we are working with community. And so how can we make sure folks understand the work that goes into the design and how that helps build trust that we are keeping their data safe? We think about generalizable. We think that this is the concept of, does this reflect the population that would actually benefit from this intervention? And so, many moons ago, we didn't study women in clinical, many moons ago, still currently, that was a bit of a joke. We didn't include women in clinical trials because, they were hormonal and that was complicated. Well, the reality is that women got sick and responded to treatments differently. And we end up in this situation where we have, diagnostic criteria that don't necessarily reflect both sexes. And so when we have a clinical trial or designing it, we want to be able to ensure that we are trying to maximize representativeness. It is never going to be that you are going to have a fully representative clinical trial, despite all of your best efforts. And this is because there are just limitations on who can participate in trials and the time and cost associated with it. But it is important to have these conversations up front with your trial team about how you can maximize that representativeness. And after the clinical trial, how can you communicate whose data was included, whose was it, and what might be helpful for the next people picking up this question in order to inform making their designs more representative to the patients in front of them. And that last concept is really about reproducibility. And so it's important that trials are reporting what they are doing because when clinical practice guidelines are made, our job as during evidence synthesis is to grade the quality of the study, to say this was a good study, this was a bad study, this study, it's completely unclear if it was good or bad. And so we need to know. You need to tell us what you did in order for us to be able to assess that. So if methods are not clearly reported, even if they were immaculate, they're going to receive a lower grade during evidence synthesis because we are not mind readers. And someone picking up this trial 10 years from now may not, have access to be able to ask questions about, your randomization strategies. So it's really important when we publish trials that we're including enough information so it's clear how the trial was designed, whose priorities informed it, how there is trust in these findings so that we are not leading to research waste. And there are many, many, many clinical trial guidelines that are available on the Equator website. This is not just about clinical trials. The Equator website has lots of great reporting guidelines for all kinds of research studies. But I would say as you are writing up your study protocols, this is a great place to start in terms of resources, because if you know what they're going to want to see at the end, you can make sure that you clearly outline your thought process for that at the beginning. So there are many different frameworks to assess like the quality of a publication. But when I'm asking myself, like, is this a good study? I'm asking myself, like, whose perspectives and what evidence informed this question? Was the design appropriate to answer that question? So is this an implementation trial of something that's never shown to be feasible? Or is this a rational approach for why they're doing the trial? Is it clear to me how risks were mitigated and how they maximize safe inclusion? Is there enough information to replicate the study? And how were trial challenges and changes made or presented? And I think this last one, oftentimes publications have word limits. So, there's not a lot that goes into the discussion usually about the challenges of operating a trial. But for me, I think it's important to take note of if there are changes throughout the trial. How did the team document those changes? Changes happen and are allowable and are encouraged in trials if they are justified. And so, we're not looking for a trial that had no challenges or no changes. I don't know that those ever exist. We are looking for a trial team that's accountable and that is transparent about when they have made changes and why. So just the last couple of points about where to get some more resources, and then I can take some questions. So there's actually a great video, and it's 9 minutes, so I won't share it today, from UNICEF here about an introduction to randomized clinical trials. They have lots of great videos, and UNICEF, sorry, not, I'm not sure what I said, but UNICEF also has some great videos out right now around good data governance for children's data. And some other interesting resources that may be of interest for this audience. In Canada, most of us who are at universities are a member of a network of networks. So Network of Networks, or N2, is a nonprofit incorporated alliance of Canadian research networks and organizations. And they have our institutions pay a membership fee to N2 so that we can access their standard operating procedures and their training for clinical trials. So if you are at a university, almost all of them have memberships. You can check on the website, and you can access training for free. So most of your institutions will have like a representative for N2 that you would have to reach out to access this training. But you can email N2 and they can tell you who at your institution can give you the right login information. And this is really important because this is a national Canadian sort of umbrella organization, and they really they do a great job of making sure that all of the content they have is up to date with all of the international standards. So I want to say that they're even a little bit ahead of Health Canada in terms of what they have to offer. So that's a great place. And of course, I couldn't leave today without talking about Impact a little bit. So, Impact is a clinical trial training platform, and we are now just about to start our third cohort in April this year. And this training platform is focused on clinical trials with children and pregnant people. So, we have three things to offer. One is our free self-paced courses and webinars. We have a one-year fellowship program with salary support, and this is to turn ideas into trial designs. And we also hold an annual meeting to build networks and innovative designs. And we have core competencies. So our fellowship program really focuses on engagement. So they are all assigned a patient and youth and parent mentor to help them build their own skills in engaging during the design process. So they gain skills in trial design. They actually design their own trial conduct and in trial communication. And so these are all available on dual code, and you just register with an email and they are free. So you do not need to be involved with Impact to access our courses. We also partner with awesome groups like CanNRT to offer fellowships. So Danielle and Ishnor were two of our CanNRT fellows. And these, in our first two cohorts, we are opening another cohort in August. So we really are proud of our partnership with CanNRT and our shared goal of increasing the number of folks who are trained and designed and trained and ready to conduct clinical trials in NDDs. So our website is impacttrials.ca. Feel free to poke around on there. We will, like I said, we are starting our cohort 3 will start this April, and in August, cohort 4 applications will open. And these are open to PhD students, to postdocs, and to early career faculty. So a couple of final thoughts. Clinical trials, they are prospective, they are rigorously planned ways to evaluate health interventions. Good trials are well-informed, they are trustworthy, they are generalizable, and they are transparent in their design and conduct. Clinical trials take a village, but they play a really important role in improving health outcomes for people with neurodevelopmental disorders. And there are additional opportunities to learn about clinical trials throughout your career. So, don't hesitate to reach out, whether you are at the beginning of your career or somewhere in the middle, in between. There's never, the wrong time to learn more about trials. So feel free to reach out if you have any questions. And maybe I'll just stop sharing my screen, Eliana, so I can see if there are any questions or anything else folks would like to chat about.